are basically molecular on-off switches. They're molecular on-off switches. They have an active state. When they're active, they have GTP bound to them, and they have an inactive state when they have GDP bound to them. Going from the inactive to the active state requires GDP to be exchanged with a molecule of GTP. So to go from inactive to active requires that you exchange the GDP with a molecule of GTP. So GDP is a lower energy state than GTP, right? Because GTP has 3 phosphate groups, guanosine triphosphate, and GTP has only 2, guanosine diphosphate. So it's easy to see that the active form of the G protein will have the GTP bound to it, whereas the inactive form will have the GDP bound to it. So if you have a G protein that's in its off form, then to go to the on state, it needs to do what? Have that GDP exchanged with GTP, right? Have it exchanged with GTP. Excellent. Oh, yes, a question.
One addition of the phosphate group is
Oh, very good. Yes, I can see. That's an excellent point. What? Go ahead. Yes, that's a very good point. In order to activate the G protein, we have to exchange the GDP to the GTP. Basically, we kick out the GDP and then put in a GTP in exchange. Now, why does that happen? Well, it probably has to do with the energetics of the cell, the energetics of the cell. It's probably a lot more easier for the cell to simply exchange it with a GTP rather than to phosphorylate the GDP while it's bound, because GTP is, for example, created in a process like the citric acid cycle. Remember, there's a stage in the citric acid cycle when a GTP is produced. When you have to rephosphorylate, for example, ADP back to ATP, you have to go through a long process known as what? Cellular respiration, right? You have to go through cellular respiration in order to generate the electron transport chain, in order for chemiosmosis to occur so that ATP can be regenerated. And where does the energy come from? For cellular respiration, glucose. So similarly, energetically speaking, it is a lot more efficient for the cell to exchange the GDP so that the GDP can then be kicked out, rephosphorylated later, and then come back in. Rather than phosphorylating it right there. The GDP is probably phosphorylated, for example, in the citric acid cycle, things like that. So GTP is used for a lot of other purposes too. So a GDP can then be rephosphorylated and then transition somewhere else, depending upon the needs of the cell. So they are just very complex ways in which the cell regulates its energy needs. And I would imagine that the answer has to do with energetics. It has to do with how the cell manages its resources. Because we learn about, in biochemistry, we learn about all these pathways. And we sort of think that each of these pathways are occurring independently of each other, but that's not necessarily so. The cell has to regulate how much it performs of each pathway, the level at which each pathway occurs according to its needs and the number of resources available to it. So that's why cellular energetics is a complex concept. And that's where I think the answer lies, why it actually has to exchange it when it could rather just phosphorylate it right there and convert back to GTP, is that the GDP can then be kicked out and then maybe transferred to a pathway where it will actually be phosphorylated again so that it can come back in. Now, how does it go from active to inactive? We need to answer that question. That's a very important question because that has to do with a problem that we see when a mutation in the RAS protein occurs. I believe that was the next question or so. When a mutation in the RAS protein occurs. So, once the G protein is on, once it's in its active state, it will proceed to activate other proteins, activate other target proteins. We'll talk about that in just a moment. But the point we'd like to understand is that once it has been converted to its active state, we would like to ensure that eventually it goes back to its inactive state, right? Isn't it appropriate to say that if we turn on the light, then we would like to eventually turn it off in order to conserve energy, right? Exactly. Similarly, the cell, if a G protein is on and it activates certain pathways, then we would like to ensure that eventually the cell turns off those pathways. However, if the cell does not turn off those pathways, that may lead to some tragic consequences. For example, cancer. So we'd like to understand how exactly does the G protein go from its active back to its inactive state. Well, here is where we have to talk about how G proteins use what they have is their GTPase intrinsic activity. Intrinsic activity. What this activity is that the G protein is capable of autonomically, meaning by itself, breaking down GTP to GDP and an inorganic phosphate. It's capable of doing that on its own. So it's a subunit in the G protein. This is a subunit in the G protein. And I'd like to go ahead and mention this right now, that a point mutation, this is an important point to know for the final exam, a point mutation in a very important G protein called the RAS protein, called RAS, a point mutation deactivates this intrinsic GTPase activity. This intrinsic GTPase activity. So a point mutation in a very important G protein called RAS, which we'll talk about, deactivates this intrinsic GTPase activity. What RAS is responsible for doing is that when RAS is active, it activates cell division. But when RAS is inactive, when it's off, no cell division occurs. So in certain instances, we would like to activate RAS, and other instances, we would like to turn off RAS. In some instances, we'd like to turn on the light, and then when we leave the room, we would like to turn off the light in order to conserve power. But if we leave the light on, that's not good either, because we will be, of course, wasting energy and money. So similarly, if the cell does not manage to turn RAS off and it remains active, what do you think that could lead to? Cancer. Because what cancer basically is uncontrolled cell division, uncontrolled cell proliferation. So we will talk about how the cell manages to prevent cell division from occurring, and what happens when something goes wrong and then cell division does occur uncontrollably. That's what we would like to learn about next. So let's see if we answer this question. What effect might a mutation in the RAS gene have? Well, we just answered this question. Right? What would it lead to? Uncontrolled cell division, right? Uncontrolled cell division. control cell division. Go ahead, sir.
All of this is occurring inside the nucleus.
This is actually occurring more in the cytoplasm. In the cytoplasm, yes, sir. In the cytoplasm, that's where the RAS is located. And eventually, when RAS, so you asked a very good question, sir. When RAS is activated, it itself will activate a whole pathway that consists basically of a phosphorylation cascade pathway that eventually culminates in the activation of transcription factors inside the nucleus. So the pathway will begin in the cytoplasm, and then it will eventually culminate inside the nucleus, where once those genes are activated that are responsible for cell division, then you'll have the effect. Because cell division has to be preceded by nuclear division, right? So mitosis has to begin in the nucleus. Then the cell, cytoplasm, divides. So that's something to think about. Any questions this far?